Welcome back, biologists. It's Mr. Forberg here, and today we're looking at another short free response question from the 2019 exam. So let's get started. It says a researcher is studying patterns of gene expression in mice. The researcher collected samples from 6 different tissues in a healthy mouse and measured the amount of mRNA from 6 genes. The data are shown in figure number 1. So the first thing that I want to do is I want to look at figure number 1 just to get a feel for what's going on there. And of course here I see that we've got those 6 different tissue types. Right? We have 6 different genes that are being transcribed and eventually translated, and our key indicates the amount of mRNA that's present in each of those tissue types. And so, for example, if I am in a clear box, there's no mRNA, there's probably no protein synthesis for this particular pathway in that tissue. All right, if it's shaded, it could be a moderate amount, so I'm making and expressing some of this protein, but not a whole ton of it. And then the very darkened in is that high amount of mRNA, and that indicates that this is a very active gene that's being actively transcribed and translated because the protein's needed. All right, so we've got a good feel for what the table tells us or shows us. Let's get a look at part A. It says, based on the data provided, identify the gene that's most likely to encode a protein that is an essential component of glycolysis. Provide reasoning to support that identification. So let's figure out what are those task verbs that we really have to focus on. The first thing that we're going to do is we're going to identify the gene, all right, that's encoding a protein specific to glycolysis. And that's a really key piece of information to focus on. What is glycolysis? Well, if we recall back to our cell energetics unit, we know that glycolysis is the first step of aerobic cellular respiration, or any cellular respiration for that fact, or for that matter. Now, why is glycolysis being singled out? Well, we should recall that all cells need to carry out glycolysis because it's going to eventually lead to the generation of ATP. So the citing of this particular process leads me to believe I'm looking for a tissue type, or I should say a gene that's actually expressed in all tissues, because all cells need to carry out glycolysis to produce ATP to carry out cell functions. So let's go ahead and see which ones are going to work and which ones we can rule out. All right, in gene E, and I'm going to start right up here in just a quick moment. All right, I see that I have a lot of mRNA expression in the liver, heart, and brain. However, I have no expression of mRNA in the kidney, pancreas, and skeletal muscle. And I know that those particular tissue types require ATP. They all do. So because we have certain tissues that are not expressing this mRNA, all right, gene E, not a possibility. Gene F, on the other hand, liver, heart, brain, and kidney, but again, no mRNA expression in the pancreas and skeletal muscle, which means we're not getting any ATP production if we're not running glycolysis. Gene F, you're out. Gene G, I see high levels of expression in the liver, brain, and skeletal muscle. I see moderate expression in the heart, kidney, and pancreas. It is expressed in all tissue types, so for me, I'm looking at gene G, but I'm going to run through the others just to make sure. Gene H, we don't see any expression in the heart or the liver. That's a problem. The same as with gene I. And then gene J, it's not expressed in any of these, so let's get rid of that one real quick. So taking a look at my data table, I'm going to confidently answer this has to be gene G that's responsible for this particular protein. Now, provide reasoning to support your identification. This is going to be my second task verb. All right, in order to do so, I'm going to support that gene G is in fact the gene that we're looking for, because all cells need ATP. Therefore, all cells carry out glycolysis, or all tissue types. Moving on to part B. Part B says the researcher observed that the tissues with high level of gene H mRNA did not always have gene H protein. Provide reasoning to explain how tissues with gene H mRNA can result in no gene H protein. All right, so I try and think about, well, what's happened successfully and where are we getting this hanging up? All right, and so we know that if we're thinking central dogma, all right, central dogma says we're going to go from DNA down to RNA, and we're going to produce some protein. Now, what we've done well, all right, we've produced this RNA here. So we've successfully transcribed that DNA. What we are having a hard time doing is we're having a really hard time getting it to this protein stage. All right, so what could be going on wrong? Well, we could have that RNA is sequestered inside of the nucleus, so we're not properly transporting it out. Maybe we couldn't add on that 5' cap to export the mRNA. All right, we could be experiencing RNA interference out in the cytoplasm itself, and so that could be locking onto our mRNA and blocking ribosomal attachment. Or we could even have a mutation within that mRNA in that maybe we're coding for a stop codon prematurely, and that's why we're not getting this fully functional protein product itself. Any one of those would be completely reasonable. Well, let's use a different color. Reasonable explanations or reasoning to explain how we could have high gene H mRNA but no gene H protein. I hope that helps. Thanks for tuning in. Have a wonderful day, and we'll see you next time.